All right, so today we're going to go over the viruses and the microbes and agents list. So first we have bacteriophage T4. So this is a lytic phage, so that means it undergoes the lytic cycle of replication, as we previously mentioned, where eventually the replication of the virus leads to a lysis of the cell and release of many, many virions. And it also has a double-stranded DNA genome. And just recall that, remember, phage just means a virus that specifically infects bacteria. And it's one of seven what we call T phages, so it's T1 through T7 phages that infect E. coli. And it also has the pretty standard phage structure that we'll look over on this next slide. So there's a bunch of components here, but I'll just go over the main ones. I mean, we kind of talked about viral structure already, but specifically for phages, right, we have the capsid, just like in the other viruses we saw, which is made of protein, and it contains the genome here. There's also a base plate and spikes at the base here, which help the phage attach and then kind of inject its DNA. So let me get my pen here. Right. So through this, the DNA will pass through this tube here and then get injected into the cell. And then, of course, the tail fibers will help with the adhesion and for the phage to find specific places to inject its genome into the bacterium. So when talking about T4, it's good background knowledge to have that bacteria naturally have a system of what are called restriction endonucleases. These are enzymes that specifically cut DNA, and they have recognition to specific sequences of DNA. And a lot of times those sequences are found in phages, which kind of shows that this system of DNA cutting proteins evolved as a way for bacteria to cut up DNA that came from phages. So it's kind of like an immune system for the bacteria. Very simple immune system. And actually, Cas9 is also a restriction endonuclease, and of course Cas9 is involved in the CRISPR-Cas9 system, where you can do gene editing using this system that we will talk about in the future. So stay tuned for that. So what bacteriophage T4 does is it makes this alternate form of cytosine C in DNA. So it makes what's called 5-hydroxymethylcytosine, just means it has this extra OH with an extra carbon sticking off of the normal cytosine. And that kind of makes it so that these restriction endonucleases can't cut sequences that have these altered Cs within them. So it's kind of an evolutionary back and forth, and actually some bacteria now have evolved restriction endonucleases that can recognize this base as well. So like it's kind of a back and forth between the phages and the bacteria, just like evolving in that way. As we mentioned, T4 follows the lytic cycle. Of course, review here: attachment, penetration, synthesis, assembly, release. You should have those drilled into your brain. Just keep memorizing that. And you see that here, and it's pretty self-explanatory what each step does. So everything else is standard here, but we'll talk about the attachment and penetration steps because there's a little more specific molecular components here. So we saw that the phages have these LTFs, these long tail fibers, right, sticking off of the end here that help them recognize where exactly they need to inject the DNA into. So for the T4 phage, these long tail fibers, what they do is they recognize specifically lipopolysaccharide. Recall that this is found in the outer membrane of gram-negative bacteria, and of course E. coli is a gram-negative bacterium. These are those things that stick off the outside of the membrane, and also OMPC, or outer membrane protein C, which is a channel protein, right? A protein that, if you've seen those diagrams, if this is the membrane, right, it lets things into the cell through this channel. So it can bind to either of these in order to help mediate the injection of the DNA. So this is kind of when the phage knows that it needs to inject its DNA. And there's kind of a model of how the phage does this. It's kind of wandering, it's almost walking along the cell surface with these long tail fibers until it finds a suitable injection site, as you can see on the right side here. And interestingly enough, so this is just kind of a tangent, but I thought it would be cool to include this. So there's been research into T4 based systems of delivering therapeutic genes or molecules to human cells. So viral using it's called AVV, artificial viral vector. If you can look into this if you're curious. So we can this has already been used for a lot of things, including like the COVID vaccine, right? So basically you take the structure of a virus and then you strip away all the like infectious quote unquote bad components, then you can kind of stick in what you want. So like you can stick in, for example, something that'll help you kind of recognize a different virus when it comes in, like the like the COVID vaccine basically is where you can put a virus in that is not infective, and it teaches the cell how to make the spike protein of the coronavirus, and we'll see that in a later slide. So I'll stop talking about that. But yeah, so here essentially we're using the capsid of the protein shell component of the T4 phage only, and you can kind of take away everything and then stuff inside a bunch of different DNA, protein molecules, etc. And then you can even stick stuff on the outside. Then you can put a lipid coat around the entire thing, and then that'll help it that when you like kind of inject these into human cells, right, they'll fuse with the human cells and deliver all these things very well. So pretty interesting. Okay, next we have bacteriophage lambda. So this is a temperate phage this time, so it can undergo both lytic and lysogenic. It can switch between the two. And it has, again, a double-stranded DNA genome that also infects E. coli, just like bacteriophage T4. So, and it was actually heavily it was one of the most common model organisms in the late 20th century, just because of this switching back and forth between lytic and lysogenic cycles. It required very tight, very organized gene regulation. So it was well studied to understand the mechanisms of gene regulation in general. So it has a very complicated system of switching between lytic and lysogenic growth, which is driven by what's called the CRO repressor, and essentially DNA damage or environmental stress can cause the bacteriophage to start doing the lytic cycle when it's already in lysogenic. We're not going to get too much into this mechanism unless it shows up on a test or something. So because it's extremely complex and hard to understand. So what we're going to do is we're going to focus on instead how the DNA gets into the cell first. So bacteriophage lambda, it's different from T4 in the fact that it's non-contractile, which means that it can't inject its DNA. Like T4, it actually needs to kind of find an opening in the membrane first, and then it can kind of slip the DNA through that opening instead of just stuffing it through the membrane there. So what it does is it'll recognize LAM-B, which is a similar to OMPC, but it's a porin protein, which again is used for import. In this case, it's used for the import of maltose, which is a disaccharide of two glucose molecules bonded together. So normally the E. coli bacterium uses this porin protein to import that sugar and get energy from it, but in this case the bacteriophage is binding to it and then stuffing its DNA through that opening in the membrane here. Okay, so there's this thing called rolling circle replication that both of these phages do, but we're not going to get into that mechanism yet because it's a state national topic, but I just want you to know that that is where rolling circle replication will come in later on. Again, here's just some more basics about phages in general. So phages, they can be cultured on what are called bacterial lawns. So lawns are essentially where you get an agar plate, and then you put your bacteria, you spread them evenly across the entire surface so that there's even bacterial growth uniformly across the plate. And then you can introduce your phages, and then what happens is that as the phages grow, you get these plaques. They're called plaques, which are these just like kind of ring-like areas where all the bacteria have died off, and that's where you can tell that the phages are doing their thing there. And it's kind of similar. It's like kind of the inverse of, right, for bacteria, you have colony-forming units that arise, whereas for phages, you have these plaque-forming units that instead are like just empty instead of like colonies. So it's like, yeah. And then this isn't super important here, but just wanted to slip it in case you see something like it. So it's a growth curve except for phages. So when we do growth curves for phages, we measure the amount of extracellular, what are called virions here. So if a phage is inside a cell infecting the cell, then it's not counted within the count of virions. So essentially, right, you start with good number of phages. You put them on the plate on the lawn. Then they all go inside, right? They infect all the cells. So your number is going to drop first after inoculation in what's called the eclipse stage. Then, of course, you're going to have a low count as the virus is replicating and making more copies of itself within the cell. And then, of course, when the cells lyse due to the lytic cycle, then you're going to have this big exponential increase of the number of extracellular virions. And, of course, wherever this peaks off is what's called the burst size, which is you can kind of calculate how many copies of the virus were made within the bacterium that you infected initially here. And before we get into the non-phages, so the viruses that infect humans, now we're going to do a brief review of the Baltimore classification system, which is just a method of categorizing viruses by the way that they get to the mRNA stage before they make proteins. So you have 7 groups here, then you have these 6 and 7 are special because they're what are called retroviruses, which means that they go from RNA to DNA, right, breaking the central dogma. So normally we have DNA to RNA to protein, as we've learned many times. But what these retroviruses do is they go backwards from RNA into DNA, as you can see here, through what's called reverse transcription that we'll talk about later. So, and then these positive, negative symbols, right? The positive just means that the genome is essentially the same as the mRNA. So the initial genome that's injected is equivalent to the mRNA. It just goes, and then this negative sign is for the complement, so the matching pair. So, right, if you start with a negative, then if you get the matching pair of that, then you turn into the positive here. So if you start with the positive, you have to go to a negative first, and then go back to a positive, or you can just go directly from positive to positive here. And let me know if that doesn't make sense, but it's not too important. And also good to know that this is called positive sense, and then this is called negative sense. All right, so let's get into our first virus, which is HIV, or human immunodeficiency virus. It's enveloped, so it has a lipid covering around the capsid, the protein shell, and it's also a type 6 virus. So it is one of the retroviruses that we mentioned here, and it's specifically this class of retrovirus, where it has a positive sense single-strand RNA genome, and then it can convert that to DNA, and it'll eventually insert that DNA into the host genome. Right, so how it does that is the enzyme that catalyzes that process is called reverse transcriptase. So normal transcription is from DNA to RNA, reverse transcription is from RNA to DNA, and of course it inserts into the host DNA, as I mentioned. And HIV is the causative agent for what we call AIDS, right, acquired immunodeficiency syndrome in humans. And then this is minuscule detail, but there are 2 types of HIV, HIV-1, HIV-2, but HIV-1 is kind of the one that we're always talking about when we say HIV, because HIV-2 is like it's much less common and not as important in this case. So, I mean, we kind of looked at the cycle of viral replication already in my virus lecture, but specifically for HIV, there's this, they've divided it into 7 more specific steps. So those are binding, fusion, reverse transcription, integration, replication, assembly, and budding here. So the important ones here are the reverse transcription, of course, because that's key to the HIV life cycle, the retrovirus life cycle, and then and then this integration is also pretty important because we're actually putting the DNA into the host genome. So HIV, of course, it can be transmitted through various bodily fluids and from mother to child as well. Important to note that it actually can't be spread through like coughing, sneezing, etc., or physical contact. It's more through specifically bodily fluids like blood. So so once HIV comes into the body, right, what it does is it has an envelope protein, so a protein on its on its membrane, kind of sticking out. Like if you've seen pictures of COVID, right? Same idea. So it has envelope protein GP20 or GP120, and what that'll do is it specifically binds to CD4 receptors, and these CD4 receptors are located on what are called CD4 plus T cells. And if you've heard of T cells, these are very crucial immune cells within our body that help communication, and they help recruit other immune cells to just help, just maintain the normal functions of the body. And then, so the virus is specifically infecting these immune cells, which is, as you can imagine, that's where the name human immunodeficiency virus comes from, because when you infect the immune cells, your immune system is going to tank and get really downregulated. So, and then there's also two other receptors that are called CCR5, CXCR4. These are just co-receptors that are also help mediate the fusion of the viral membrane into the host membrane, as you can see on the left side here. So you have that CD4 initially binding, then you have this co-receptor here that helps it fold and make this whole process and fuse the membranes together. So after binding and entering the cell, HIV, of course, it uses the reverse transcriptase enzyme that we talked about to turn its RNA into DNA. And then once it does that, this DNA will get localized into the nucleus of the host cell. And then it uses this protein called integrase, which, as the name suggests, it helps the DNA from the virus integrate into the host genome, as you can see on the left side. And then once the viral DNA is inside, it's called a provirus. And you can imagine, as the cell divides, as it's doing mitosis, right, it's going to replicate its DNA, and along with the normal DNA, it'll replicate the viral DNA as well. So the viral DNA will get carried along with the rest of the host genome as the cells keep dividing. So eventually, like you can imagine, a large portion of your body, or at least of your T cells, will get infected with the virus, and then bad things will start happening from there. So, and of course, the provirus that's in the DNA serves as the template for the transcription of viral genes. So it's the same process. So you have the viral DNA in the genome, which gets turned into RNA, mRNA, and then you make the viral proteins. And then what happens is that as those viral proteins are synthesized, they get kind of they get inserted into the host membrane, as you can see here, these green structures here. And then it'll kind of pre-create this kind of envelope and capsid structure already. And then all the virus has to do is stick its genome into the middle here, and then it'll bud off, and then kind of pinch off part of the host membrane and create these fully functional HIV infectious particles that are now outside the cell and can go and infect new cells as well. And that process is called budding. So there's kind of two terms when you have HIV in your body. The first is just HIV infection, which just refers to the presence of HIV within your body. And then you also have AIDS, and AIDS is specifically diagnosed when your CDC4 plus T cell count, right, the cells that HIV infects, when that cell count drops below 200 cells per millimeter cubed. And of course, you get a blood test and measure this. So whenever you drop below that threshold, you're officially declared that you have AIDS. So and symptoms include a fever, headache, muscle ache, rash, cough, weight loss, kind of some flu-like symptoms as well. And of course, like we mentioned, the immune system, right? The immune cells are being attacked by the virus, so the immune system will get severely weakened, which means that your body will now be more susceptible to outside attack from other pathogens. And then HIV infection, of course, it can't be cured, but it can be treated. And the most standard treatment is what's called antiretroviral therapy, or ART here. There's I think 7 or 8 different classes of drugs that can be used within this therapy, but some of them include reverse transcriptase inhibitors, right? You're stopping the reverse transcriptase from making the RNA into DNA. You can also use fusion inhibitors to stop the virus from fusing to your host cell membranes. You can also use integrase inhibitors so that the DNA from the virus doesn't get inserted into your genome. So just a few examples of ways that you can restrict different parts of the retrovirus life cycle. Okay, next, measles virus. So measles, it's a enveloped. Again, it has a lipid covering around the capsid. It's group 5 this time, which means that it's negative sense ssRNA, single strand RNA. So it is the genome itself is the complement of the mRNA that's going to be created to make the proteins later. And of course, it causes measles. And it's known for being highly infectious, specifically between children, and it is only infecting humans here. Oh, and that reminds me, HIV actually is kind of evolved or derived from a similar virus in chimpanzees, I believe. So just a cool thing to know. Yeah. Measles is spread through droplet transmission, through sneezing, through breathing the same air as someone that's infected with measles. So of course the viral particles will enter the lungs, and then what happens is they're taken up initially by these immune cells, which are called macrophages, which we mentioned in the bacteria lecture. So remember, macrophages, they're going to come up and phagocytose, right? They're going to eat up the virus and absorb it within their cell body, and dendritic cells will also do something similar. And then over time, right, the virus will spread to other immune cell types and eventually to the rest of the body and even the brain, which can be extremely lethal here. It has two different surface proteins with on its envelope here. As you can see, the first one is a fusion protein F, and then the second one is hemagglutinin, which is denoted as H protein. And these are the two proteins that help mediate the virus's fusion with the cell membrane and allow it to enter. So these envelope proteins. And then the specific role, the hemagglutinin is the initial thing that binds, while the fusion protein is the thing that allows the membranes to fuse specifically. And then there's 3 different receptors that these proteins can bind to and recognize to enter cells. So first you have CD46, which is present on all cell types. Then you have CD150/SLAM, and this protein is specifically localized on immune cells, such as the macrophages and dendritic cells here. So these are going to affect immune cells. And then Nectin-4 is present in the epithelial cells. So remember, epithelial refers to the outer lining. So for example, in the trachea, right, those would be your, like the outermost, right, as soon as you, like if you stuck a finger in your throat and then you touched around, those would be like the epithelial cells. So this receptor is present in the placenta and the trachea. So, and you can kind of see it in this graphic how, right, initially the H protein binds to the receptor, and then these fusion proteins kind of open up the membrane and allow them to fuse together. And the life cycle, pretty standard to what we've seen in the past. And then, of course, the fully formed virus particles, once they're translated and then assembled together, they can exit the cell via budding. And so measles, it's pretty common among children, especially those that aren't vaccinated, because there is a simple fix to the spread. It's just getting vaccinated, getting the measles vaccine. So symptoms include fever, cough, runny nose, and then the most kind of standout symptom here is the red rash, the classic red rash that you see here. And of course, it binds to that CD150 and affects immune cells, which means that it'll also weaken the immune system to some degree. So talking about the vaccine, it can be either administered by itself, or the more common one that you normally see is is the MMR vaccine, or the measles, mumps, rubella. Mumps and rubella are also viral diseases that are similar to the measles virus here. And what the vaccine does, it contains a weakened form of the virus, and essentially that trains the body to be able to recognize the real virus once it's there. And that's an example of what we call a live attenuated vaccine. So live meaning the virus is still active, and it's attenuated because it's been kind of stripped down, it's been watered down a lot, so it's not infectious or dangerous. It's solely what it does is that it trains the body to create antibodies that can now be used to recognize the actual measles virus whenever it infects the body for real. And then the alternative to a live attenuated vaccine is an inactivated vaccine. I'm just telling you guys about the second type of vaccine here that we'll mention later. And this contains viruses that, well, actually, I don't think we will get into this for this lecture, but it's just good to know about this. So it contains viruses that are essentially dead. So they're just sitting there and they can't replicate, and your body just accepts that and then uses that as the model for the way to produce immune responses for an actual virus infection. Okay, next, SARS-CoV-2. You guys all know this one, right? It's the one that causes COVID-19. It is enveloped, so it has a membrane as well. And this time it's a group 4 virus, so just positive single-strand RNA. It's also found in bats, and it's a part of a group of viruses known as the coronaviruses, due to the classic kind of spike protein structure that they have around their envelope here. So as we know, the virus can be spread through droplets from person to person, through sneezing, coughing, breathing, etc. And then how it enters the host cells is, excuse me, it's spike protein, or S protein fuses with what's called the ACE2 receptor. And when it does that, it can either first be internalized, as you can see on the left side, it can first be internalized within an endosome or a lipid kind of lipid kind of bubble within the cell, and then the genome can be released, or it can directly fuse with the membrane and then release its genome immediately as well. And it specifically targets the airway epithelial cells, so such as those in your trachea, right, in your breathing pathways, the alveolar cells, which are in the lungs, and then also the vascular endothelial cells, which are just the cells that line the inside of your blood vessels here. So not too much to talk about here. Again, the standard, right? The replication, you get the genome in, it replicates, you synthesize the viral proteins, you assemble them, then you release them out, this time through exocytosis. So this is slightly different from what we saw before. One second. Yeah. So this is actually, well, it's basically the same thing as we saw before, but it's just what the process is called. Exocytosis is when you have a vesicle containing the virus, and then the vesicle itself fuses with the host cell membrane and then releases the contents within, which is the virus here. So so let's talk about COVID-19, the disease caused by SARS-CoV-2. This is actually pretty important to know because people kind of use the term coronavirus, COVID-19, SARS-CoV-2 kind of interchangeably, but COVID-19 specifically refers to the disease that's caused by SARS-CoV-2. So and it stands for coronavirus disease 2019. And then the symptoms, we know all of these, so don't have to review that. Then we can talk a little bit about how the specific vaccines work, because there's 3 different general mechanisms here. The first is an mRNA vaccine. You've probably heard of this. So it delivers mRNA that codes, this should be S, that codes for the spike protein of the virus. So when you deliver this mRNA into the cell, right, the ribosomes in the cell are going to translate that mRNA into the spike protein, and then the cells will they'll insert the S protein onto their cell surfaces, right, and then those those spike proteins can then get recognized by immune cells that are floating throughout your body, which will create antibodies and then allow you to it trains you to recognize the spike protein the next time that the body sees it, which is when you might actually get an infection. So, and then something similar can be achieved through what's called a viral vector that we kind of mentioned before, right? The AVV thing with the T4 capsid, and this is called a vector vaccine. So here, essentially we take what's called an adenovirus. It's just a specific type of virus. In this case, I believe it's like a chimpanzee adenovirus specifically. So you strip away all the bad components. You're left with this kind of shell that you can stick stuff inside of. And the virus is still active, except it's been re-engineered to carry the instructions to make the spike protein. So now when you get infected by this kind of neutral virus, right, you're going to start expressing the spike protein. Then the same thing happens as with the mRNA vaccine, where now you're making the antibodies for the spike protein, and then you can defend your body against it the next time you encounter the virus. Then finally, you can, so these, right, you're inserting something that will eventually create the spike protein, but you can actually just insert the spike protein itself as well. So you can use protein subunit vaccines that carry the spike protein and then evoke the same immune response that we mentioned here. And this is pretty cool, right? You can isolate, or you can get the gene or the DNA that codes for the spike protein. You stick it on a plasmid, as we've kind of alluded to in the past, right? With the insulin, the production of insulin. And I don't think everyone was there during that lecture, so that's okay. So I can go into more detail in that later, because we'll actually talk about specifically how insulin is manufactured using these plasmids that are inserted into bacteria. But the idea here is that you take the spike protein gene, you stick it in a bacterium, and the bacterium can mass produce that spike protein for you whenever you feed it. And then you take away, you purify that protein, then you can stick it in a vaccine, and then you inject that in a person. Okay, next, smallpox virus. So smallpox, it's also, the official name is variola virus. It's enveloped as well, so it has a membrane. This time it's group 1, so double-stranded DNA virus, and it causes, of course, smallpox in humans, right? Smallpox is one of the deadliest viruses that has kind of riddled mankind for the past few thousand years. So very important virus to know about. And it's actually smallpox is the only human disease that has been essentially completely eradicated. So the only last natural case of someone getting it, like from the environment or from another person, is in 1977. Basically, there was this massive vaccination campaign that covered like almost all countries, and then that massive vaccination campaign led to basically the complete eradication of the virus in the wild. And we'll talk about some of the implications of that in a later slide. And it's played a very, very significant role throughout history, such as, like, I'm sure you guys have learned about, right, when the European, like, conquistadors, right, the Spanish conquistadors arrived in the Americas to explore and to take, all the valuables and stuff. They carried smallpox with them, and that actually like completely devastated the Native American populations at the time, and that's only one of the major effects that this virus has had in the past. So very historically significant. So there's two types here, variola major and variola minor. And a variola major is the one that is the most common, the most severe, and the one that we will be talking about for the next few slides. So smallpox infection, it occurs through contact with infected bodily fluids. So for example, right, the smallpox, if you've seen pictures of people with smallpox, they get these kind of sores, like these like kind of like blister-like things over their body, and those blisters release pus, so exposure to that pus can cause infection, or you can get it through droplet transmission from sneezing, coughing, air sharing, etc. So variola is actually incredibly complex, and it's a very large virus. So it has over 200 different proteins that it encodes for within its double-stranded DNA genome, compared to, for example, HIV, another common virus, which has only 15 proteins. So, and I was actually researching the mechanisms of how this virus gets into our cells, and I couldn't really find that much conclusive evidence there. I'm not sure if that's because I wasn't searching hard enough, or if it's just not very clear at the moment, but yeah, so didn't really have much to say there. But we do know that DNA viruses, most DNA viruses, they replicate within the nucleus where the rest of the DNA is of the cell, whereas this smallpox virus actually replicates in the cytoplasm, which is slightly unique. And part of this, the reason why there isn't much literature on this, might be because of something that I will mention in the next few slides. because the research on the smallpox virus is extremely limited. First of all, right, because we eradicated the virus in the wild essentially. So there's only 2 facilities in the world that still have active samples of the virus. And these facilities are extremely heavily guarded and placed under many a legislative control, essentially. So there's actually been a lot of debate regarding whether these samples should still be kept or if they should just be eradicated. And the people that want the people that want to keep the viruses are saying essentially that using them could be beneficial for not using them, but like, having them could be beneficial for conducting research that could help with the study of other pox viruses, like for example, monkeypox, which has been, I guess, in the news lately and on kind of a resurgence, while others warn that it could be too dangerous to have these, and if they're not properly handled, right, if, let's say, some breach happens and like the virus gets out somehow, right, that would be disastrous for the population, especially because they stopped vaccinating people in the mid 70s. So that would be really, that'd be really, really bad if someone happened to like bioengineer smallpox or something, which is another big concern that people have debated over. And actually in the late 70s, someone who was researching the virus actually contracted and died from it. So I guess that lends further credence to the idea that we should probably destroy the virus, but that's a whole ethical debate that is not, I guess, important for our discussion today. But so in the 1900s alone, in 100 years, smallpox caused nearly 500 million deaths around the entire world. And that's just 1 century, right? This has been literally, this has been plaguing humans since ancient Egypt, since like there's records of mummies that have like smallpox and stuff. So, but just a little history about the vaccine, the original vaccine. Okay, so technically, before, even hundreds of years before this, people in like China, India, and then near, I believe, like Iran or something like that. Yeah. So they were already had similar treatments that they had developed, but Edward Jenner was kind of the first guy to make a formalized vaccine and the first person to kind of scientifically document what was going on with this vaccine. So what he did was he noticed that there was a similar disease to smallpox called cowpox, which was infecting cows and humans. And he noticed that basically people who had contracted cowpox, which is a lot less severe than smallpox, if they had already contracted it, then they were kind of essentially immune to smallpox. So what he did was, this is kind of disgusting, but he took, right, there's like, the blisters. He took the fluid from the blisters of people who had cowpox, and he injected that into people in order to vaccinate them for smallpox. And this actually worked really well because the viruses are similar enough to where if you inject the less severe cowpox in, then the body recognizes and is essentially trained how to fend off the smallpox virus as well. So yeah, that worked, and then that's kind of where this whole vaccine idea came about, and then it developed, and then now of course we have a much more developed smallpox vaccine, and this time we use what's called the vaccinia virus, which is very similar to smallpox, and this time we use this instead of the cowpox to kind of render resistance within cells. Yeah, so the vaccine that uses this vaccinia virus, this is actually a live attenuated vaccine, like one of the categories we mentioned before. So the active vaccinia virus is injected into the upper arm, and essentially, right, so it's crazy because it leaves these like really characteristic scars here, and the reason of that is because you basically, for the vaccinia, you have to inject it several times in the same area over and over, and then once you do, in that region, the vaccinia virus is still doing its thing. So it kind of forms this like sore and like this like blister just in the region where you were injected. And then over the course of a few days, it kind of bursts and then it goes away. So over those few days, your body is fighting off the vaccinia virus and then learns how to defend against the smallpox virus. And like I said, the vaccination stopped in the 1970s after the eradication of the virus here. And of course, this trademark scar, you might have seen it maybe on your parents' arms or just adults in general who were growing up in the 70s, and that is the trademark sign of the smallpox vaccine. And that's it.